Salam sejahtera to all viewers. I am Arniza Ghazali from Universiti Sains Malaysia, Penang campus, and the lead researcher of this study, together with prominent team members from USM and various private sector. I'm sharing the outcome of a study entitled Bio-hindrances to Fibrillation of Lemongrass Leaves, SQ for Big Data Analytics. This would be the flow of the presentation. I would briefly introduce the project, briefly state the current issues, the objectives, method, share the outcome, and conclude the findings. Let me briefly introduce the project, which places the main interest on lemongrass biomass or Cymbopogon citratus. Worldwide, leafy biomass encompasses some 30 million tons of plant waste. Still in the current practice, post-harvest of lemongrass bulb, lemongrass leaves are normally left on the field despite possessing commodity such as therapeutic cellulose and valuable ground tissue. This creates wastage and a host of unnecessary environmental issues. Therefore, the team attempted to define a better management plan to productively manage leafy biomass such as lemongrass leaves. In such an attempt, lemongrass was wedded with a smart alkaline peroxide pulping system, which was developed in the US in 1989 as a solution to the polluting pulp and paper industry. The system is eco-friendly in that it is sulfur-free and chlorine-free, and the outstanding advantage of the system is that it is a rapid system producing high pulp yield or fiber yield depending on the form of feedstock introduced to the system. As for lemongrass leaves, this means that the biomass is reacted with per-hydroxy radicals, and the next section would explore the possibilities of and the criteria of the product produced from this system. The effort points to the three main objectives of the study, that is, to examine the outcome of the alkaline peroxide mechanical pulping of lemongrass, to project a new application of the generated product, which was initially thought to be pulp mass, and recommend big data analytics as plant waste management plan for informed manufacturing. And this is particularly important as we pave the journey to Industry 4.0. To obtain the initially hypothesized pulp mass, lemongrass leaves was collected from the field, washed, dried, and downsized before subjecting to the APMT fibrillation concept. Attempt was also made to produce lab hang sheet, and a detailed microscopic analysis was performed to understand the nature of the generated mass. Let me now present the outcome of fibrillating the fresh lemongrass leaves or the dried green leafy biomass. Advanced microscopic analysis shows intact vascular bundles having between 17 to 19 micrometer width. The unfibrillated structure suggests that the generated per-hydroxy radicals was consumed in some way but not in fiber fibrillation. Apparently, therefore, the unique lignin-retaining reaction between per-hydroxy radicals and the biomass was hugely hindered, suggesting at the same time the recalcitrant tissues composed of lemongrass leaves. In contrast to the outcome of fibrillating the green leaf biomass, the rusty brown lemongrass leaves obtained as a result of prolonged sun drying shows evidence of rupture. A close-up look at it shows evidence of vessel-free bundle sheath, suggesting invasion of cellulose degrading microorganisms, probably the lytic polysaccharide monooxygenase fungi. Besides that, biochemical degradation also occurred as a result of prolonged sun drying, resulting in chlorophyll degradation to phyllobulin, causing the rusty brown of the sun-dried biomass. The evidence of microbe attack or fungi attack of the biomass during sun drying was also shown by the presence of hypha protruding from one pit to another, as shown on the micrograph. At another site of rupture, there were also evidence showing the presence of microcomplex strong interlayering tissues, which was the potential biohindrance of radical penetration into the structure. Besides that, evidence of nanopitted structures suggests another possibility of hindrance factor to radical penetration, and these are biohindrances to fibrillation of lemongrass leaves. Examining the heavily ruptured bundle sheath further shows the presence of a partially ruptured vessel structure, which exposes crystal druses of about 1 cubic micrometer. These were characterized as predominantly calcium by the energy-specific analysis of X-ray interface to the scanning electron microscope, suggesting the calcium-loaded nature of APMP pulp of lemongrass leaf biomass. Besides the presence of calcium druses, fine fibers were also found as heavily embedded in the non-cellulosic structure. The complex recalcitrant nanopitted sheath, the hardly liberated fine fibers, together with the evidence of heavy load of calcium druses, all point to the fact that lemongrass leaves are no suitable candidate for paper making. At this stage, the findings project the potential use of the calcium-loaded leaf biomass to bioglazing application as preliminary attempt to polish several surfaces shows glazing effects with minimal emission of dust, which promotes process safety by eliminating the source of respiratory hazard. This is an example of surface, initial unpolished surface, lemongrass polished surface. The findings therefore signal the need to triangulate biomass types, to process, and applications. In a productive manner, not only to intelligently manage plant biomass, but also to enforce informed manufacturing that matches all product to specific application. This rules out the need to discard outlier, as every process outcome is properly triangulated to an opportunity of useful application. And this can be enabled by big data analytics. On a micro scale, the identified bio-hindrances directed the generated mass to a specific functionality. The outcome signals the need for big data analytics as a systematic management tool that maps every process outlier to a specific other use, thus widening the possibility of zeroing failure and wastage, making past failure a current and future opportunity. With that, I thank you all there for being present throughout this presentation. Stay safe. Assalamualaikum warahmatullahi wabarakatuh.